my name is Kurz, and tonight I will be talking to you about vaping. So please raise your hand if you or someone that you know vapes. Well, for those of you who are either lying or really don't know, vaping involves the use of an electronic cigarette to vaporize addictive chemicals such as nicotine. These liquids often contain many chemicals to give them fruity and savory flavors. A study conducted in 2017 found that 19% of high school seniors had vaped nicotine. This same study found that 21% of high school seniors had vaped flavoring chemicals only. Both of these numbers are up over 10% from the previous year. So why are so many adolescents using these potentially dangerous products? A study in 2016 illustrates that while they may have some understanding of the harm, they really have no firm understanding of what it could do to their bodies. Participants in one study believed that vaping was equivalent to smoking water. The lack of understanding regarding these chemicals and the overall usage increase led to many research projects. A study in Colorado found an increase in airway diseases in humans who used electronic cigarettes. Another study found a high correlation between cracked and broken teeth and electronic cigarette usage. The chemicals used to flavor electronic cigarettes have been found to cause cell death in the trachea and lungs, leading to many issues. These same chemicals have been found to cause oral facial deformities in frog embryos. This leads us to my project. While nicotine has been adequately studied, the chemicals used to flavor electronic cigarettes have not. My study aimed to answer the following question. What effects, if any, does exposure to diacetyl, 2,3-pentanedione, and acetone have on mouse behaviors? So in order to see if these chemicals are having any effects on their behavior, a true experimental design was utilized. This meant that in addition to the 3 flavor treatment groups, I also had a control group which received no chemicals, that was used as baseline and comparison. For my study, 32 of these mice were used. This is an ICR strain, which is an albino outbred line. They're said to be very docile, but take my word for it, they bite very hard. I used 32 of these, and there was 4 males and 4 females for each testing group. So all of my procedures were pre-approved by the Institute for Animal Care and Utilization Committee at the University of Colorado Boulder. This means that I was trained by a registered nurse, a veterinarian, and 3 of the facility employees before I was allowed anywhere near the lab. Before this project, I had no idea how many precautions go into ensuring the safety of everyone involved in research projects. So the 3 chemicals I chose to use were diacetyl, 2,3-pentanedione, and acetone. In a study conducted by the Harvard School of Public Health, these were found to be the 3 most common flavoring chemicals used in the most popular flavors vaped by adolescents. Unfortunately, mice don't really like to vape, and they don't make electronic cigarettes small enough for them to do so. So twice a week, I administered these chemicals to the mice's water. After the 3 weeks of exposure, which was all of their adolescent period, behavioral studies were conducted using the elevated zero maze, which we have up here on the screen. The elevated zero maze measures anxiety-like and risk-taking behaviors. As you can see, it's broken up into 4 quadrants. These 2 are considered closed because they have walls, and these 2 are considered open because they do not. So increased time spent in these areas is going to indicate increased anxiety, and increased time spent in the open areas is going to indicate increased risk-taking behavior. After my study was complete, I was able to determine that each mouse gained weight within the average for the strain, indicating healthy adolescent growth, and each mouse also stayed hydrated for the entire time of my experiment. This meant that each animal could be included in my final results. So after running statistical analysis on each variable from the elevated zero maze, 2 of them really stood out, and the first one is time in closed quadrant 1. So to get to closed quadrant 1, the mice had to cross an entire open quadrant. This is because each mice begins their experimental testing period in closed quadrant 2. Now, crossing a little bit of plastic may not seem like a big deal to us, but to the mice, this is regarded as very risky. As you can see, mice in all 3 of the chemical treatment groups chose to make this cross, while none of the 8 control mice chose to make this cross. This indicates that the mice exposed to any of the 3 flavoring chemicals were more likely to cross an entire open quadrant than the control group. The second variable that stands out is open entries. An open entry is when a mouse completely enters an open quadrant. Its hind feet and tail have to be completely past the opening. So as you can see, there's not a lot of difference between these groups, but this is usually a very constant measure in all behavioral studies. So my 3 chemical treatment groups do have higher means than the control, indicating difference there. So what does this mean? Overall, my data does show that mice exposed to flavoring chemicals in my study were more apt to take risks than those not exposed. This was determined using these 2 variables and a multitude of others from the elevated zero maze. After finding these results, I began to wonder why this may be happening. Adolescents already engage in more risk-taking behaviors than adults, and this is due to an imbalance of brain activity in the prefrontal cortex, which is that area up in yellow. This area is still developing until we are 20 years old, and it is most responsible for risk assessment. So, if the mice in my study were exposed during all of their developmental period, and the flavoring chemicals they were exposed to were inhibiting the correct development of this specific brain region, it would exhibit as increased risk-taking behavior. This is constant with prior research that diacetyl may be altering homeostatic pathways in mice brains. My data presented and already published literature together concludes that acetone, diacetyl, and 2,3-pentanedione may be altering brain biochemistry and correct development of the prefrontal lobe. So, an important takeaway from my study is that electronic cigarette flavoring chemicals, specifically acetone, diacetyl, and 2,3-pentanedione, may be altering brain biochemistry, increasing risk-taking behavior in adolescents. This could be very dangerous for adolescents who already engage in an increased amount of risk-taking behaviors. Though my data is certainly interesting, this topic urgently needs to be further researched for the safety of all users. Before walking into AP research in the fall, I had no idea that I would be spending countless hours in a collegiate lab surrounded by such amazing teachers, students, and professors. I had no clue that my one small idea would turn into such a big project that took multiple months and taught me so much. Overall, this process really taught me that no matter how many setbacks I may face or how many people may think that my dreams are too big, if I believe in myself and truly work hard, I can accomplish anything. I hope that each of you will continue to ask questions, wander about the world around you, and stay curious. As the wise Albert Einstein once said, the most important thing is to never stop questioning. Thank you.